I'm here with Jeffrey Brinker, Distinguished Professor at UNM. You are on the forefront, basically, of a revolution in medicine. We're talking about examples of nanoparticles that can actually destroy cancer cells.
The nanoparticles that we make, which are, get this 100th or 1,000th time smaller than a cancer cell, one particle loaded with a particular cocktail of drugs can kill one cancer cell. So it's an amazingly efficient ability to kill cancer with these particles. And the whole concept is that we want to deliver drugs directly to cancer cells and nowhere else in your body. I think everybody is familiar with chemotherapy, where you just inject pure drug into your veins and the drug goes everywhere. And it kills cells that tend to reproduce very fast, like cancer cells, but also your hair, right? So everyone goes bald, this sort of thing. So what we're trying to do is put all the drugs in a tiny particle that can go directly to the cancer cell of interest and kill that cancer cell. And the center of that particle is essentially a sponge created on the nano scale, okay? And that sponge, if you put it into a beaker with your drugs, it will suck up the drugs into the sponge. And this sponge, these particles that we make, just if you can think about this, so if I were able to unfold the nanostructure within them, I would get a carpet with 1 gram of powder. I would get a carpet 10 meters wide and over 100 meters long with 1 gram of material. So this internal surface area sucks up and absorbs all these drugs. So that's sort of on the inside. But on the outside, we disguise the particle to allow it to not be recognized as a foreign invader by your body. And we put particular types of molecules on it that are recognized by the cancer cell surface.
And sort of accepted by it.
Right. And so we can direct these particles to interact with the cancer cell surface. And through this interaction, they get taken up actually into the cancer cell where they release their cargo. So it's kind of a Trojan horse from the standpoint on the outside, it looks friendly to the cancer cell, but once inside the cancer cell, we have a surprise.
The cancer cell doesn't know what it's in for. Tell us about nanoparticles.
Why do we use nanoparticles? Again, they're very small relative to the size of the cancer. And that smallness is really important for several reasons. First of all, the small particles have to circulate in your body, okay? So you inject it into your body. If the particle is too large, it might be viewed by your immune system maybe as a bacterium or something, and it would get cleared out of your body. And if it's really too small, it might go through your renal system and go out in your urine. So there's a particular size particle that most efficiently can enter the cancer cell, because without getting the drug inside, again, you don't kill it, okay? And then there's also penetrating a tumor. So that you all familiar with the primary tumors of a cancer. So you want this particle small enough that it can work its way into the tumor and access all of the cells within the tumor. And then another really serious problem of cancer is not just the primary tumor, it's metastasis. So what we want are these small particles that can penetrate everywhere and hopefully find and seek out and destroy these metastatic cancers.
It really is minuscule beyond belief.
I can't just point to them because they're so small. We have to look at them with very powerful microscopes. But a nanometer is defined as 1/billionth of a meter. Wow. 1 × 10⁻⁹ meters. And that's just, you can't even think about things on that scale. These particles that we're using are 100 or 1,000 times smaller than a typical cell in your body. And so, and they're of the size of molecules, right? So what we want to do is we want to have the interior of this particle, this sponge, if you will, have a pore size that just the right size to accommodate a drug molecule.
there must be some challenges with the types of equipment that you have to use to even study this.
There are two aspects of this. One, the equipment that we need to look at these particles is very expensive because we have to use what's called electron microscopy. But I can make these particles with very simple techniques. And so the one major way that we make the particles that we're using, these nano sponges, if you will, is to do something just like, I think everybody's familiar with a vaporizer, right? So you can plug it in and it creates little liquid droplets, okay? And so what we do is we make liquid droplets, and within those liquid droplets we put particular chemicals that when the droplet evaporates, the chemicals are programmed, essentially pre-programmed to form this nanostructured material. So it's simple. It's called self-assembly. So this is fantastic. Like my graduate students, right? You walk into the lab and what are they doing? They're sitting around, oh, we're doing self-assembly, right? So the particles assemble themselves.
Wow.
So we developed this extremely robust way to have the particle nanostructure form by itself. So that's very easy for us, that part. And then once we form this sponge, we do something else that's pretty simple. We put on the outside of this sponge what's called a lipid bilayer. So every cell in your body is covered by this lipid bilayer. It's a very thin membrane. And so we put that over top of this sponge. So we load the sponge with our drugs and we put this membrane over it to keep the drugs in. And then on the surface of the membrane, we put molecules that recognize a particular cancer. So the particle is kind of stealth-like. On the outside, it doesn't get recognized by your body's immune system, but it has a few of these what we call targeting ligands that will recognize the cancer cell. So when it comes in contact, it will interact with that cancer cell and then go into it. And then we have the Trojan horse effect occur.
How do you get them to do what you want them to do once they're in there?
And we do that by putting molecules on the surface of this particle. And we identify these molecules by kind of a fishing expedition kind of thing. So we make a library of a whole bunch of very small, what are called peptides. These are just very small amino acids combined together, like a very small protein. So you can imagine if we had a huge library of all kinds of random peptides like this, and we sort of dip them into the solution with our cells, right? Mm-hmm. And we sort of see which ones stick and which ones don't. And then, so we identify peptides that only stick to the cancer that we're interested in. And so we use that peptide that we identify, we put it on the surface of our particle. Wow. And it will go in and, it doesn't get directed toward the cancer, but if it encounters the cancer, it will start to bind to it. And this binding leads it to get internalized or taken into the cancer cell. So we have two really expert women in our lab, Carly Ashley and Katie Epler, who are experts at identifying these ligands. And it's a process that you go through, but it's very robust process, and that's how we kind of train the particles to sort of do what we want them to do.
How far away are we now from, say, the human clinical trials with this research?
The research that we're doing now, a very important part is to take it into clinical trials and ultimately humans, of course. And so what we have is we have a grant that's funded through the National Cancer Institute. They have an alliance for nanotechnology and cancer. So I've teamed up, I'm a sort of a nanomaterial scientist, but I teamed up with Cheryl Willman, who directs the UNM Cancer Center. And so her interests are, of course, combating cancer in human patients. In particular, what's called acute lymphoblastic leukemia, which is it's a childhood leukemia and one that can have a very low outcome. And so we've teamed up to make particles that will be useful to treat this acute lymphoblastic leukemia they refer to it as ALL. So we put these molecules on the outside of our particle that recognize these ALL cells and can enter them and kill them. So in terms of how quickly can we get this into human trials, there's a progression. First, you have to show that you can kill cancer in a petri dish. Okay.
Start there.
And then you have to show that you can kill cancer in a living organism. In a mouse model would be typical. And then if you can show that, then you might go to a primate, and then finally, into a human trial. The shortest would be sort of a 5-year period. That would be a very rapid, taking something out of the lab, getting it into a human clinical trial. So the FDA would have to approve this. So there are two critical things. First of all, it can't do you any harm, right? It doesn't mean that it has to really cure you, but if it has any kind of toxicity, that's a huge problem. So we have to prove that these are very, very safe. And then we have to prove that they have some benefit, right? So they help you to some extent.
What other applications and nanostructured materials can you imagine going forward?
Nanoparticles are being explored, throughout the world for other types of application, for sustainability, for us to have a sustainable environment. We really need to focus back on solar energy, and so these particles can also be used to convert solar energy into electrical energy, right? So this is they can be used in, for example, photovoltaics.
Sure.
They can also be used to help to convert chemicals from one type to another. So you do this all the time in your car. You have a catalyst that will convert CO that's created by your engine into CO2. Okay, so very small. You have a catalytic converter in your car probably, and that has nanoparticles of different metals in that case. So these are used as catalysts.
What inspires you to do this?
This porous sponge that I had described to you, I made this back in 1999. We had a paper in a famous journal called Nature that described our ability to make these sponges, okay? And I had at that time the idea that these might be useful if we could load them with drugs and use them for drug delivery. You can't just load a particle with drug without kind of sealing it inside. So it was only more recently I was thinking about these membranes that I was mentioning. So we put a cell membrane over the outside. So we were interested in understanding how do cell membranes work, and one way to study them is to support them on something and study them with a number of different physical techniques. And so I was thinking, wow, we're making cell membranes, we're also making these porous materials. Could I put the cell membrane on the outside to seal the cargo in? And that was sort of the inspiration for making this particle that we actually call a protocell.
What's the next breakthrough?
We feel that pretty soon there'll be personalized medicine, right? So now, the human genome has been sequenced. So I'm thinking, in the future, you're going to have on your driver's license your genome. And there might be certain types of medicines that are just perfect for you. So personalized medicine, right? And in these particles, like I said, they're very versatile. So we feel that we could put any combination of these drugs and make them personalized for you or someone else. And so this is, I think, one of the future goals. And of course, you can build more, there's a lot of information you can build into even a nanoparticle. So they might be able to, you might have a GPS system in them to see, where are they in your body, this sort of thing. So all these kinds of ideas, I think, are not too crazy.
What are the downsides to nanoparticles and some of the things we've been talking about?
If you build a car, I can go in and inspect every little part of the car, no problem. And so now you have this particle that you can't see. And you just have a beaker of it, and there's millions and millions of these particles in there. And how do you assure yourself that you've got what you think you have? And how do you prove it to the FDA? So you have to develop techniques that will assure you, or a way to kind of judge what you have. That's a tricky part of this process. And then, of course, cancer itself, people have the idea that cancer is a thing, but cancer can take multiple forms. Even the same type of cancer can have different subpopulations, and whether you can kill subpopulation A, but maybe you don't touch the other one. And so to really, ablate cancer, destroy it, you have to kill all of the cancer cells, and that is a very tough problem.
Fascinating stuff. Thank you so much for joining us today to talk about this.